Welcome to OmicsBox, Bioinformatics Made Easy.
Okay, so my name is Miguel Ferrando. I am part of the Biobank team, and today we are going to perform a complete genetic variation analysis in Vitis vinifera, a plant species, with particular focus on Genome-Wide Association Studies using OmicsBox. In this session, we will review how to prepare raw GWAS data to carry out a comprehensive pipeline, variant analysis pipeline, to obtain high-quality genetic variants, focusing mainly on single nucleotide polymorphisms. We will then track down those variants to specific chromosomes and associated to phenotypes of interest. At every step, we will visualize the results, especially the GWAS results, using different plots such as Manhattan and QQ plots. The data that I picked for today comes from a paper published in 2022 at Oxford University Press, and it is publicly available at NCBI Bioproject for genotype data and also phenotype data, which can be accessed through this specific repository. As a quick description of the dataset, the organism is Vitis vinifera, commonly named as Grapevine. It is the plant cultivated for table grapes and wine. For this study, the DNA was extracted from young leaves and then sequenced through genotyping-by-sequencing with Illumina, following a RADSEQ protocol. The layout comprises both single-end and pair-end short reads, with a total of 628 samples across almost 1,000 FastQ files. So it is a large dataset, and it tries to capture the genetic diversity of this species. The reference genome that we will use is the latest version telomere to telomere V5 assembly from Grapevine Genomics Encyclopedia, and the phenotypic data has been curated to include only a subset of traits under the same conditions. This analysis is quite complex and it requires the integration of multiple tools. So I have standardized an end-to-end OmicsBox workflow, which consists on 7 main steps and is divided in 3 parts. The first part is the data quality control, followed by variant discovery, and finally the association analysis. First, we need to evaluate the quality of the sequencing reads and try to maximize their quality for the analysis. Then we will try to identify variants and we will try to detect those variants through different steps, starting with the DNA-Seq alignment using a reference genome to generate a BAM file, BAM files with aligned reads, and then these aligned reads are required for BCFTools for the calling step. We will then apply a filtering to only retain the quality variants. And finally, we will curate the final BCF file to recover any missing SNPs and use that file to do the association analysis. We will also study the population of Grapevine, population structure, and do the association of genotypes and phenotypes using GWAS through statistical models with CAPIT3. All of this will be done using a graphical user interface in OmicsBox and no need for coding skills. And we will also use cloud computation for some of the tasks. So I will now switch to OmicsBox and we can begin with the analysis. Okay, so now once we are inside the application, the first thing we need to do is the quality assessment of our raw reads. So general tools, FastQ tools, short read quality assessment with FastQC. We input our reads first, and then we can add any additional adapter or contaminant sequence if we have them. This is not the case, so we will skip this part and then click on run. I have the FastQC ready. So I'm just gonna load the report, the results. And then we have this table with the results ready for review. And the table contains information regarding the dataset. So we have sample names, total number of reads per sample, read length, GC content percentage. And if we want to get the results, we have to click here in the summary report side panel. And if we expand this report, we have a table of overall results with different evaluations for each sample. All of these results must be interpreted given the context of the library and sequencing technology. So we should expect some results beforehand, and we are not surprised by some of these fails and warnings, but we can study those in detail. So the sequence quality appears to be fine, quality scores as well, and then we have a fail flag for the per-base sequence content, so we will study that. And then the per sequence GC content is not concerning. If we go down, we have additional results, and we can see that the adapter content failed. This means that we probably have some adapter sequences present in our samples, and we also have overrepresentation and then high duplication levels. As I said, we need to consider the context of this data. The duplication levels are actually expected in this case, since we used a GWAS RADSEQ strategy for sequencing, and we might have, since this strategy only sequences part of the genome, we might have PCR duplicates, and also Grapevine. The Grapevine genome is highly repetitive, so we expected these results. We are going to study those more in detail by generating a report for this specific sample. So we have a statistics table here with the encoding, which is Illumina, total number of reads, and so on. And we will focus on the results table. The fail status appears here in the per-base sequence content, and we need to check this to see what happened, and also the adapter content. So let's check the details for this one. Yeah, we can see that the distribution percentage of bases at the beginning of the reads appears to be abnormal. So this means that we have low quality bases in all reads, and this could be due maybe to a motive for a restriction enzyme site for RADSEQ, and anyway, we will need to exclude this, and we just need to keep the other this part of the reads, right, which look very good. The sequencing went well, and this we will just maintain part of the reads, right? So we will trim sequences by position at this specific position, and then if we go back to the report and click on adapter content details, we can see that in fact we have an Illumina adapter present in our sequences. We check the position, and we are gonna cut this right here and remove both adapters and low quality bases through pre-processing with Trimmomatic. So let's go back to general tools, FastQC, short read pre-processing with Trimmomatic. Again, we input our reads. We will do this in single and parent batches. We can remove adapters automatically if we know the specific adapters present, or even add adapter sequence files. Other parameters which can be configured, and then we are going to uncheck this option. And the second configuration includes an edge trimming manual option. We will use the length trimming in this case. So we will remove so first keep bases from start, 5' N. We will keep the first 55 bases. So if we go back here, let's do something else. I think this looks much better now. Let's go back to Trimmomatic. And then if we use the edge trimming, length trimming option, if we keep bases from start, we can keep the first around 55 bases to remove the adapters. And then the second step will be to remove bases from start, from 5' start, which will be the first 5. And then another configuration with quality trimming in case you want to trim by quality. And also we will filter by average quality and so on. We specify the output reads folder and click run. So now our sequences are now clean and they are ready for the next phase of the analysis, which is the variant discovery phase. In this phase, we first need to generate to do the mapping, DNA-Seq alignment, and generate a BAM files for the calling step. So we will go then to genome analysis, DNA-Seq alignment, and then we have two different strategies, BWA and bowtie. BWA was used in the original paper and is actually very accurate for when mapping against a large reference genome. But as an alternative, we have bowtie. In this case, we're going to stick to BWA. We input the reads in single end per n and the reference genome in FASTA file, in FASTA format, sorry. And again, we have a configuration wizard with the algorithm parameters, and we can fine tune this by minimum seed length, bandwidth. I'm not going to go into detail here since we are going to keep this by default. I want to keep the pipeline as standardized as possible, and the parameters are actually very fine for this specific analysis. So if you need any additional information on any parameter, you can just click here and read the information, the message for each one. And we're just gonna see this the overall parameters, which are the scoring parameters for matching score, mismatch penalty, and output BAMs. And then just destination again, click run, and this will take a little bit. So we are going to open up the report directly to see the results. And first we have some input information, first regarding the reference with the sequences and lengths, and our sequencing data used for the alignment. We have all of the reads, number of libraries used. In this case, a subset was used for this analysis. And then we have single end, which was the one used, and then the FastQ format, right? The results table includes total alignments per sample, map percentages, duplication rates, and so on. So The mapping percentage is very high, and this is a very good sign. So the alignment worked very well. And there is also a high duplication rate, which was expected. The duplicates will be removed in the calling step. So I don't think we should spend more time in this one. I'm going to plot the alignments. And we can see the alignment percentages for each sample. And in green, we have the map percentages, which are for all cases above 90%. And this is the perfect indicator that we can proceed with the next step, which is the variant calling. And so we have now generated our BAM files with the aligned reads, and we have a good quality of alignment. And so we can switch to the genetic variation module and then click on variant calling, and two algorithms to choose from. First, we have BCFTools and FreeBias. BCFTools is actually more recommended for diploid species, and FreeBias for haploid or polyploid. Since we have a diploid organism, we're going to choose BCFTools. It was also used in the reference paper, so we're just going to stick to this one. And then we will use the BAM files just generated with BWA, and then the reference genome, which is actually required for this step to do the variant calling. We will remove duplicates as a BAM pre-processing step to lower the duplication. And then we have some basic MPyLab parameters which we can adjust by mapping quality, maximum depth, and so on. These parameters are within the expectations, and so we're just gonna keep them this way. We have other advanced MPyL parameters which are more complex, and I don't actually recommend adjusting this unless you need to. So we can just continue, and then we will generate a BCF file. Let's go to the report. Okay, so this is the variant calling report from BCFTools. First, we have our input data with the reference, and then the number of BAM files for all the samples. We have as results a first table of total number of SNPs and total number of indels. We have a very good amount of SNPs to start. These are above 3 million. And we are going to focus on this for the GWAS. The frequencies for different number of alleles are also represented here in this table. The vast majority are V-allelic, which is actually expected for a diploid species, but we also have multi-allelic variants. And this could be due to the heterozygous nature of Grapevine. But we will actually continue using this for GWAS. We have another table with the counts for the total number of variants, genotypes, which are variants times samples, and then number of heterozygotes, which is interestingly lower than that I expected, and the missing data, which is very low, and it's quite good. So we will see after filtering how these percentages change, because of course we need to check the quality. We have a good amount of variants now, but we need to check the quality of those. because we will probably have to go through a filtering step to improve that quality. So if we open up a plot for this, we have this proportion quality depth plot, and this is the quality of the variance normalized by depth. The depth coverage in RADSEC experiments it's actually low, so we expect to have not very high quality depth ratios, but we should expect ratios higher than 2, 3. So the rest of them should be excluded with filtering. The majority of the variants are in the lowest quality ratio, but we still have good amount of variance in the rest of them. So we will see how the filtering step improves this battery of variants. So we will just go to genetic variation again and click on variant filtering. We choose between variant filtering option and D-docent. D-docent is free bias specific. So let's just use the variant filtering option. And then we have some parameters here to adjust. We import our BCF file, and then we have the proportion quality counts, which should be 2 or above. Then row read depth, minimum 10, thread quality 20, average mapping quality 55. So standard parameters, which are like minimum thresholds. And then the genotypic options, we should check the remove multiple alleles. This is recommended for GWAS to avoid false associations. And then I'm gonna mention also the minimum allele frequency threshold. This specific 0.05 value was used in the paper as well, and it is actually a threshold you use to keep or eliminate rare variants depending on frequencies. So we're just going to keep it with this threshold, and then you have other free bias specific options. Not interested in that. And then we will generate a filtered BCF file. Then filtering. Report. Okay, so we have this variant filtering report now, and we have the percentage of exclusion, which is 85%. So a lot of variants were excluded in this step. We still have a good amount of 476,000 of variants. And the number of heterozygotes increased, which is actually expected for this type of organism, since it has heterozygotes. And the missing data has increased as well. This missing data will be recovered afterwards with Beagle using imputation. I'm going to open up another proportion quality depth plot for after the filtering. And yeah, as we can see, we have removed all of the variants below this value, and so we will keep variants with minimum quality for GWAS. Still have a good frequency. It lowers with quality, but still we have a good total amount. So. Continuing with the analysis, we will proceed now and do the genotype phasing and imputation to reconstruct haplotypes with phasing, and then do and recover missing SNPs with imputation. Oh, sorry, not annotation. Yes. So now we are going to use Beagle to curate the BCF file that we have just generated with filtering. And then we have a configuration for phasing, imputation, and general parameters. Both of phasing and imputation are just fine. The general parameters should be adjusted. We don't want it to estimate effective population size. We're going to use 10,000. It is typical for plants. The effective population size reflects the expected diversity within the population, and this value should be used in this case. So sliding window can also be adjusted as Beagle reads the chromosomes in windows, and we can just set like the windows in centimorgans, which are like genetic distances, and then also the overlap between windows and so on. So let's leave it this way, and then the output BCF after Beagle will be generated. And now this BCF file is now ready for the final phase, the association analysis. This is the most important part of the webinar, and we are gonna go and dive in. So population structure analysis. And now we are going to use the BCF file to study populations. This step is key to understand the biological context of Grapevine. This specific organism has a lot of historical recombination, has suffered a lot of hybridization through different techniques like grafting, and so we need to understand how these, if we have subpopulation, or how the population structure works for this species, since we will need to correct it afterwards with GWAS, and so we need this analysis as an essential step. So we will use ADMIXTURE for this using the BCF file from Beagle, and we will set a linkage disequilibrium, which is 2.9, which is the non-random association between alleles. And this parameter is actually critical in this type of analysis. LD is used for the association of SNPs. So the higher the LD, the higher the association, and it is used by GWAS to infer information about different SNPs. So we can also set a window size, which is the number of nucleotides to look for linkage disequilibrium. And then, we need to do a pruning. We need to set a maximum pruning to understand, like, we don't want, for population analysis, we need to just keep the association as random as possible to focus just on populations, right? So we can also adjust some ADMIXTURE parameters and then click run. So let's open up the ADMIXTURE results, and we will get different models with different cross-validation errors. These errors represent the how each model, how well the model explain the populations within Grapevine. So errors appear to be very similar in all cases. We can just keep the k equal to 10 for statistics, and then we can click run, and we will get a population statistics report with Tahima, heterozygosity, and so on. These negative values were not expected. Actually, the negative Tahima could mean rare variants, and heterozygosity as well is very negative. But we can actually, I think we can interpret this in a better way if we plot through, I mean, you can choose between different models and then plot a stacked bar chart, PCA, genetic distances. So we're gonna generate the three of them. And so we can start with the PCA. Okay, we in this PCA we can see populations in different colors, and we can see the percentage of admixture between populations, right? And samples. So we can see a mixed cluster here, which are like populations, I mean, individuals with different with mixed genetics. And then we also have pure clusters for population 3 or population 9. We can see that we have like subpopulations right here, right? So if we want to study this with more statistical detail, the likelihoods here represent, we have a color gradient with distant groups in blue and then close groups in red. So as we observed in the PCA, population 3 appears to be more distant from the others, and population 9, and then 3 and 5. But if we want to get a bigger picture of populations, we can just go to a bar chart, which I have right here. And then, yes, so we have the percentage of admixture for each individual sample. And each color represents a different population. And so we have samples with pure genetics, but we also have others with a high degree of admixture. As we can see here, we have like clusters. We have like a lot of samples which cluster within specific populations and have like homogeneous colors. So this could mean that we have subpopulations and we have like complex genetics architectures that we need to correct with GWAS models, and this needs to be studied in more detail, right? So if we go to the GWAS, we will use again the same BCF file as in ADMIXTURE, and then we will also use a phenotypic traits file with which needs to be a tab separated file with different columns. The first column should be the sample, which should include the sample names, which should be the same names as in the BCF file, and we need to include also other columns for the traits and the phenotypic values. So we have imported that, and now we have the configuration of GWAS. We have the SNP filtering because GWAS does another filtering step with math threshold, 0.05, and missiness threshold, and so on, right? Especially important, the LD to 0.5 to perform the LD pruning, which is a filtering of SNPs based on linkage disequilibrium. And then we can also normalize phenotype data, attach kinship matrix, covariate matrix with additional variables, and then the most important part, which is to select the GWAS model for the association. So in this case, I'm going to choose the multiple locus mixed linear model. This is an extension of the mixed linear model, but it is a more accurate to detect complex genetic architectures. So we will choose that and then click next and select the destination folder for the GWAS results. And then so we can go to GWAS and upload the results. And this is the GWAS results table. First, we have the phenotypes, we have the SNPs IDs, the chromosomes, specific positions, then minimum allele frequency for each SNP, number of samples, and then the effects. The effect values are actually very relevant here. So the higher the effect the higher the effect and the better the p-value. So the higher the influence of the SNP on the trait or phenotype. So we can, so we have this table with the p-values, adjusted p-values, and then if we want to look at the results, we can generate the summary report, and then so the GWAS report give us some information on the input data, the variant filtering summary with the final amount of SNPs used for GWAS and the samples. And we have a list of traits with associated SNPs. So for instance, we have mean cluster weight, which corresponds to the actual cluster of grapes. And we have glucose fructose, we have mean berry weight, and more of those. We are going to focus on the mean berry weight, which has the most associated SNPs, such as 4. And so if we continue studying this specific phenotype, we can go to charts and then genotype information, and then generate a PCA for mean berry weight, click run. And we get this PCA for this specific phenotype. We have a mix of all of the individuals with different phenotypic values. And I mean, we first before plotting or studying the phenotype information, we need to first see how the different individuals, what are the phenotypic values on each specific individual, and if they cluster or not. So they are all mixed, and we don't have clusters. So there's no like interferences of like populations or anything. So everything's mixed, and this means that we can proceed studying this phenotype, the phenotype information, right? And if we go to this chart and to this specific option, we can select again mean berry weight, and we are going to generate a QQ plot and Manhattan plot. So first, the QQ plot will give us information on how this specific model worked for the phenotype trait. No, sorry. Yes. Okay, and so we see that we have the observed values and expected values. We see a good fit of observed p-values with respect to the expected. The expected would mean that there is like a random associations but we can see a long tail of different p-values, which can mean that some SNPs are actually correlated to this specific trait. So now I think we can go and finish with the Manhattan plot. And in this Manhattan plot, we have the different chromosomes represented by different colors, and the SNPs that fall above or below a p-value threshold of significance. So we can actually see 4 statistically significant SNPs in 4 different chromosomes, right? So first one is in the chromosome 1. Then we have another one in chromosome 13, another one in 17 and 18. So we have located those SNPs within specific chromosomes as discussed earlier. So I think I'm happy with this final plot, which is actually very cool and with a lot of colors. So I think we can go back to the presentation and conclude the analysis. So I think as conclusions of this webinar, we successfully conducted a genetic variation analysis pipeline from raw reads to GWAS. We characterized subpopulations and the percentage of admixture within this panel. We identified genetic variants correlated with agronomic traits. And then we map those variants associated with a specific one, which is the mean varied weight. Actually, these studies can go way beyond this analysis, and you can do enrichment analysis to study specific annotations and integrate that information as well. But I think this is a good starting point for other downstream analysis, such as enrichment analysis, as I said, with tools like Magma. And yeah, so finally, this I hope this analysis helped. I'd like to thank you all for your time and attention, and I hope we had a lot of insights today.